Welcome to our video on Chapter 21, Genome and the Evolution. To start off, we'll talk about genomics, which is the study of many genes and how they interact with each other, as well as proteins. This was a gateway into bioinformatics, which uses computer science and data management to organize genomic research. This eventually was the reason for the Human Genome Project. And so the Human Genome Project began in 1990, and it was to find every gene in the body and see what it does. This gave the science community a bigger understanding of the purpose of human genomes and resulted in the complete nucleotide sequence of each chromosome. And scientists had different approaches to start the project, but the best one was the whole genome shotgun approach, which starts with the cloning and sequencing of DNA fragments from randomly cut DNA. The computer programs then assembled the short sequences into a single continuous sequence. This picture shows how the DNA starts out as whole, and then it cuts into little short pieces, and then it repeats itself, and then it copies and continues to copy throughout. So this project was influenced as an approach called metagenomics. So metagenomics is where DNA from an entire community of species is collected from an environmental sample and then sequenced. The Human Genome Project also used linkage maps, which map the location of different genes on the chromosome. They also use physical maps, which mapped exactly where every chromosome was and the exact number of each chromosome. So the entire process of the Human Genome Project was basically gene annotation, which is the process of identifying the locations of genes and all of its coding regions in a genome, as well as determining what all of the genes do. So gene annotation is important because once they learned of the number of sequences, they could find out which represented the site where transcription factors would bind to control gene expression, and it eventually determined how those sequences are in the promoter of specific genes.
So I'm going to talk about concept 21.3, genome size. Genomes vary in size, number of genes, and gene diversity. This means that a gene size is not particularly equal to its gene in a particular organism. So when comparing the three domains, bacteria, archaea, and eukarya, there's a difference in gene sizes between a eukarya and a prokarya. Most bacteria genomes are known to have between 1 to 6 million base pairs. The size of genomes for archaea are generally close to the size of those of bacteria. Eukarya genomes are larger, so basically in most animals and plants, they have at least 100 base pairs. For example, as shown in this picture, there are 165 base pairs to a fruit fly genome, while humans are known to have 3,000 base pairs. About 500 to 6,000 times as many as a typical bacterium. Besides telling whether or not something is a part of a certain domain, that comparison of genome sizes doesn't really reveal any relationship between the genome size and the organism's phenotype. The number of genes also varies between the domain that an organism belongs to. So bacteria and archaea have fewer genes than a eukarya. Much bacteria and eukarya have from 1,500 to 7,500 genes. So this means that the number of genes in at least 40,000 or more eukaryotes are more than those of archaea and bacteria. Some wonder how a human can function the same as a nematode if it has the same amount of genomes as a nematode. This is because the vertebrate genomes in humans get more bang for their buck from their coding sequence because of an extensive alternative splicing of RNA transcripts. For concept 2.4, talks about how only 1.5% of human genomes code for proteins and or RNA. The rest of DNA in the human body, also known as non-coding DNA, is used or was thought of as junk DNA, but that's now changing because scientists are learning more and more about this non-coding DNA, and they're talking about how they have more important functions. Both eukaryotic and prokaryotic cells have DNA that can move from one section of the sequence to another. These jumping DNA sequences were discovered by Barbara McClintock. She was looking at a pigment of maize and discovered that these genes have removable chunks called transposable elements in them. Some of the non-coding DNA in these maize can undergo a type of recombination called transposition. So these transposons, as they are called, can be cut and paste or copied and paste. The cut and paste are these sequences are cut out and pasted into another section, or they can be copied and pasted into another section. A piece of the messenger RNA can be acted on by a reverse transcriptase and plugged back into the DNA instead of being translated.
On the other hand, DNA also includes simple sequence DNA, which is when there are short copies of the same bases, such as AAA-TTT. Short tandem repeats are similar to simple sequences, but they contain two or more bases at the same time. And chapter 21.5, it talks about duplication, rearrangement, mutation of DNA that contributes to genome evolution. Chromosomes changes are the main factors of evolution and differences between species. This is the result of errors in cell division. There are many extra copies of chromosomes or mutations when DNA copies itself during cell division. The outcomes of various mutations in DNA may eventually result in new species. Scientists have conducted research in when genomes and chromosomes from different species are compared in order to see the evolutionary relationship and process. This is called comparative genomics. One of the very famous comparisons is the human and chimpanzee chromosomes. Humans are examples of species that have been created through the attachment of other chromosomes, which is from chromosome 12 and 13 from the chimpanzees. This photo from the biology book displays the human chromosomes and chimpanzees are very similar. The only difference is that the human chimpanzees chromosomes are attached while the chimpanzee chromosomes aren't. And chapter 21.6, comparing genome sequences, provides clues to evolution and development. Rearrangement is another contributor to genome evolution, which occurs when exons are mixed and matched. In addition, the movement on intransposable elements may carry a gene or group of genes in a new position. This change most likely is positive to the species. Scientists have also found what is called homobox, which provides instructions for making a string of 60 amino acids to aid in the embryonic development.
Thanks for watching!
I don't want a lot for Christmas. There is just one thing I need. I don't care